Welcome to the podcast of Hemisphere, the official journal of the European Hematology Association. Hemisphere's podcast presents insightful, expert discussions about recent hematology publications. We hope you enjoy.
Hello, and welcome to this recent episode of the Hemisphere podcast. My name is Dr. Charles de Bock, and I'm from the Children's Cancer Institute in Sydney, and I'm going to be your host today. And I'm joined by Dr. Thomas Mochet, INSERM Research Director of the Laboratory and Laboratory Head at the Gustave Roussy Institute in Paris, France. And I'm also joined by Dr. Alex Fagnon, who completed his PhD with Thomas, but is now an EHA Junior Research Grant Fellow at the MRC Weatherall Institute of Molecular Medicine, the WIM at the University of Oxford in the UK. Alex is also the first author of the paper that we're going to be discussing today, which was part of his PhD project with Thomas, and this paper is entitled "The ETO2 Transcriptional Cofactor Maintains Acute Leukemia by Driving a MIB EP300-Dependent Stemness Program." So, welcome to you both this morning, and I'm looking forward to discussing some of the main findings and also a little bit of the background to the story behind the paper. So before we get into it, I'm going to start with some context because for the audience, we just want to place a bit of background, and that one of the challenges in hematological malignancies is that whilst we understand that the ectopic expression of transcription factors are key drivers in the disease, essentially they're still undruggable. But with recent progress with the introduction of menin inhibitors, for example, understanding the higher order transcriptional complexes kind of provides a new way to drug these factors. And so in your current paper in Hemisphere, you've focused on characterizing members of the ETO family, in particular the ETO2. And so perhaps I can start this conversation by asking you to explain a little bit about the role of ETO family members, and in particular ETO2, and how they are involved in acute leukemias.
Yes, thanks, Charlie. It's very nice to discuss that. In fact, there were a lot of studies already on the ETO family members, and there are several of them, 3 of them, also called CBF2T factors. And in normal hematopoiesis, they are known to bind to other transcription factors. They are cofactors. They don't bind DNA directly. They bind to transcription factors, and they play a role in maintenance of hematopoietic stem cell, but also in differentiation, like erythroid differentiation. And in leukemia, they have been involved as partners of fusion protein in several of them, like in the AML1 ETO in acute myeloid leukemia, in ETO2-GLIS2 in acute megakardioblastic leukemia, but also in NFIA, ETO2 and ETO in acute erythroid leukemia. So in fact, they are partners of several fusion protein. And an interesting point is that they possess a domain that allows tetramerization that was shown by the group of John Bushweiler. And by interfering with this domain, using forced expression of a small peptide that was developed by the group of Christian Wichmann, you can disrupt the tetramerization of this fusion and inhibit leukemia development. So, in fact, these factors are probably big transcriptional complex making, and they can be compared maybe as aircraft carrier, where transcription factor can land or take off. And in this dynamic manner, they control hematopoiesis, and if it's altered, lead to leukemia development. So there is really a great importance to understand how they precisely work.
Yep. I love the analogy of an aircraft carrier in terms of getting all the complexes together. That's fantastic. So perhaps I can ask you, how did this project actually begin? I mean, Alex, this is part of your PhD project, and perhaps you can provide some background on how it began.
Yeah, so it began in the lab in at least through 2 different observations. One was, as I mentioned, on one of the fusion, the ETO2-GLIS2 fusion, and we were working on that, and we showed that this peptide could actually, that was developed for ETO directly or targeting ETO, could also actually work on this ETO to GLIS2 fusion. So it was showing us that we could, using this peptide, we could alter the ETO2 function. And that was the first observation. The second one was the work by Alexandre here, previous work in which we collaborated with Yorkshire, but also numerous international collaborators to actually characterize acute erythroid leukemia at the genetic and transcriptional level. And we showed that several members of this complex, GATA complex, including ETO, could be overexpressed. And that was providing the observation that we should go deeper into how it works. And that's why we developed then models to inactivate ETO2 that Alexandre will discuss more.
Yeah, that's, on that, I mean, you're talking about the inactivation, and your paper on the technical side uses a number of really elegant cellular and molecular techniques, and particularly the use of the DOX-inducible CRISPR knockout system. And, you know, I mean, I know I've tried that before, and there are probably a lot of listeners themselves who are interested in establishing this technique for their own projects. So perhaps you can give some insight on your experience of that system, because traditionally, way back in the day, before probably some of the readers even were alive, the DOX system itself was quite leaky. So perhaps you can explain your perspectives on that and the challenges and what people might want to think about before they embark on trying to establish the DOX-inducible CRISPR knockout system in their lab.
Yeah, completely. The first thing to take in consideration, at least in our case, was that we knew that ETO2 inactivation will be lethal in the cells.
Yep.
So what it means is, like, first we needed to transduce the cell with this doxycycline-inducible model, kindly provided by the lab and designed by Juanes Zuber, to first express ETO2 in the cell in a DOX-inducible manner. And once this has been done, so then we use basically a standard CRISPR screen to target ETO2. And the idea was, when we transduce the cells, basically, then we sort single cell and put this in culture, like at the single cell level, to get clones with doxycycline in the media to, in the meantime, overexpress or at least maintain the ETO2 expression in a exogenous manner, so to counteract the effect of the CRISPR in the cells.
Awesome. Yeah, I think I've also, Johannes Zuber has been extremely generous with his constructs and his information. I'm glad you mentioned him, because I think if listeners don't know about his work, I think you should definitely go and check out his amazing resources and link up with Hannes, because it's really changed the way that we can manipulate gene expression. So going back to the paper, I mean, you confirm that ETO2 at the global level is associated with both the transcriptional repressor, maybe in a more classic sense, but that you also found that ETO2 binding is enriched at active chromatin, actually that marked by H3K27 acetylation. So tell me more about this finding that you show in your paper.
Well, first, it was not a huge surprise to find genes both upregulated and downregulated, followed by ETO2 inactivation. In the way, like, pretty much in all studies when you involve like RNA-seq, you always found genes that are upregulated and downregulated. We obviously found, as you said, like more genes upregulated, followed by ETO2 depletion. So therefore, supporting previous studies that show that ETO2 is still mainly a repressor. And then concerning genes that were downregulated, followed by ETO2 inactivation, then the question was, does those genes are directly regulated by ETO2 or not? And then comes to some ChIP-seq analysis that we have done of ETO2 in both PDX, IEL PDX model and this cellular model. And one surprising thing that we showed is that actually E22 is mainly located at enhancer region. And the second thing was upon E22 inactivation, we observe that both H3K27 acetylation and K4 monomethyl at this enhancer was decreased upon E22 inactivation, that support the idea that probably E22 in a way could be positively involved in this regulation of enhancer and therefore their associated genes.
Yeah, that's I think a really key, right? Because when you see changes in gene expression, you really need that ChIP-seq data to show whether it's a direct effect or a downstream secondary effect. And I know I was going to ask this a little later, but you mentioned the ChIP sequencing, which was so key, and yet ChIP itself can be challenging. And in particular, Thomas, you mentioned that in this case, ETO2 isn't a direct DNA binder, and so yet you managed to produce some beautiful ChIP sequencing data. So again, is there any maybe you can provide some tips maybe for those also looking at doing ChIP and making sure and how to really get the best results technically for ChIP, which can be, for those who have done it, can be quite challenging to do.
Yeah, completely. We actually are using two distinct, let's say, approach that were not originally provided in the standard ChIP, at least at the time that we performed the experiment. The first one is exactly as you said, ETO2 is a cofactor, and doing ChIP-seq on cofactor that don't bind directly to the DNA is, in most cases, quite challenging. So to this aim, in addition to PFA fixation, that is basically a DNA protein cross-linker, we also use a protein-protein cross-linker named DSG that people now are mostly also using in ChIP-seq, particularly for cofactors. And the second approach that we are also used is we use enzymatic restriction MNAs to digest the chromatin instead of using a traditional sonication. And this in all ends help a lot and decrease basically the noise, or at least increase the positive and noise ratio, as well as the resolution of the peaks. So these two approaches help quite a lot to get those beautiful data.
Yeah, I think that's key, right? Because it's really nice to hear that you use the micrococcal nuclease. For those of us who are umming and ahhing about the best approach, and there are a number of sonicators, without going into too much detail here, it's great that there's two options there, especially if you don't have access to a sonicator or you're questioning whether your sonicator is actually working. But let's get back to the paper. I guess one of the key findings is that the direct regulation of the transcription factor MIB, which is actually implicated in multiple leukemias, as well as interaction that you find with the histone acetyltransferase CBP EP300. And in fact, you show that there's this large ETO2 MIB EP300 complex at these active loci. So how does this new finding help us exploit this therapeutically, given this new push to interrupt these protein interactions?
Well, first, the link between ETO2 and MIB was not described, and while the link between MIB and CBP P300 was already described as a viable target by the team of Alex Kentis. So then, basically, given that, the investigation of the interference of CBP and P300 with ETO2, at least from our side, seems to be quite interesting, at least to investigate. So then we treat the cells, the cell line, with this, with basically two inhibitor of P300 CBP. One is a pharmacological inhibitor, and a second approach was to use a PROTAC to target CBP. And what was really surprising, we were not expected that targeting P300 and CBP would lead to this degradation, this protein degradation of ETO2. So that was a quite very nice surprise to us. And this obviously leads to potential way to investigate this ETO2 CBP P300 as a pharmacological way, because like ETO2 have been described, and as you said in the introduction, as a kind of protein carrier supporting transcriptional complex in both normal and malignancies, and a quite number of oncogenes, and particularly fusion, have been shown to interact with ETO2. And I do have in mind, as an example, this TCF3 HLF fusion, and that have been described by the lab of Jean-Pierre Beauquin to be sensitive to P300, and also that have been shown to interact with ETO2. So there are link that ETO2 is one of the main and essential protein to target, even in this case, to disrupt these oncogenic transcription factors, complex and oncogenic. That could be potentially a way to investigate even more.
So just quickly expanding on that, you did mention that ETO2 also plays a role in normal hematopoiesis. Do you think there's a therapeutic window there, that there's a greater reliance on leukemia cells compared to normal hematopoietic stem cells and progenitor cells?
Well, potentially, exactly as Thomas said in the introduction, in our previous work, we also identify that ETO2 itself and some of transcription factors that have been shown to be interacting with ETO2 are even very highly expressed in at least in erythroid leukemia compared to normal erythroid differentiation. So that's why we thought that this still and potentially is a way to target erythroid leukemic cells, particularly.
And what has Alexandre shown also is that using single cell RNA-seq and comparing the same populations as much as possible, there was an increase activity of the ETO2 signature in the leukemic population compared to the normal counterpart, like normal stem cell versus. So although there will be really this question about whether and where to put the window, there could be a dose effect that we can target, but this remains to be addressed clearly.
Yeah, good. But exciting. I mean, it's a wonderful paper, and I encourage the readers to go and read the paper, because I don't want to discuss all the amazing findings, but some of the things is that the way that the paper is actually written and how we read it is not necessarily the way that the journey for the paper actually followed, and there could be some to and fro. And perhaps you can let us know if you had any really unexpected findings or challenges and surprises that you had along the way, that you went, that we should really follow that up, and to add a bit of color to the story, if you will.
Yeah, well, one really big challenge at the very beginning of the study was to get these patient-derived xenograft from acute erythroid leukemia patient. And that was really a challenge that took us a really long time. We probably started this over 10 years ago with another person in the lab. We probably engrafted like over 30 patient samples to get, at the end of the day, Alexandre was able to do a ChIP-seq on 3 of them that were reproducibly engraftable and getting enough cells to do all these ChIP-seq analysis. So this really took us a long time. but it also anchored things really into the patient side in addition to the cell lines that we had. So that was a major challenge, but once we got them, it was really nice to see also that the peptides that we mentioned at the beginning, the ETO interfering peptide, really affected and inhibited leukemia development in vivo. And that was really an important turning point to really invest into understanding how it works more mechanistically at the ETO2 level. So that was an important step for us.
I mean, so in terms of, and just to take a step back, you said, it's amazing how long these things take, like 10 years to collect the patients and engraft. And, people don't always appreciate when they see the final paper and all the history behind it. I mean, one of the challenges is with patient samples is engrafting into NSG mice to have this unlimited supply that we can study. So what is this similar to AMLs when you use NSG mice that only 30% of cases tend to engraft in mice, or have you tried other mouse models? Is that one of the reasons why one of the challenges was to propagate the PDXs or make PDXs in this case?
Well, yeah, we tried several recipients, but we found that acute erythroid leukemia is particularly more difficult to engraft compared to other AML. We had experience not with broad AML, but with acute megakardioblastic leukemia, pediatric acute megakardioblastic leukemia. And in that case, we had about 40% engraftment. For acute erythroid leukemia, we have way less. We have around 20%. But also, we have short-term engraftment. And secondary serial passage were more difficult. And it is possible that there is a component of an MDS component maybe in these pathologies that and we tried some other recipients, and it was not necessarily much better in our hands.
Okay, good to know. Well, this has been fantastic insight into some of the background. So what's next? What's the next step? I mean, people can go and read the paper, and I guess for you, this is a nice landmark, but science always continues. Alex has moved on. Congratulations on a fantastic work, of course, Alex, for your PhD. But Thomas, maybe, what's the next step?
Well, as Alex mentioned, the nice link between P300 and ETO within the same complex raise some question. At least it's known that EP300 CBP are histone acetyltransferase that can modify histones, and that's an important part of their function. The question is whether they modify other proteins and whether they modify maybe transcription factor, cofactors in this complex. And given that we see such a drastic effect on the stability of ETO2 once we use inhibitors of P300 within few hours, we can think about how this modification could stabilize probably the complex. So if we had unlimited resource, a good thing would be to identify all the post-translational modification on these transcription factors, including acetylation, to see whether these partners are acetylated or modified by other ways. So that would be of interest, but probably for the future. Excellent.
Yes, the magical unlimited funding question. So I appreciate that. We can dream, and that's fantastic. Look, both Alex and Thomas, congratulations again on your wonderful manuscript in Hemisphere. I'm sure the readers will now be excited to go and find it and read it and look at all the details. And I thank you both for your insightful discussions and also some of the challenges you had during that project. And I wish you both very well in your ongoing studies, particularly Alex, who I said again is an EHA Junior Grant Recipient Fellow now at the Oxford. So, it's great to see the EHA supporting the next generation of scientists. So congratulations.
Thanks, and thank you very much for organizing these podcasts. Very nice. Thank you.
Thank you for listening to this episode of the Hemisphere podcast. All of Hemisphere's content is open access and can be found at www.hemispherejournal.com. We hope you will join us for future podcast episodes.